I'm Daniel Levine, and this is the Bio Report. There have been significant advances to immunotherapy since their first introduction, but challenges remain to realizing their full potential in solid tumors. That's because of the ability of tumors to use multiple mechanisms to evade the immune system. Akamis Bio is developing tumor gene therapies that are capable of targeting tumors and driving expression of multiple immunologically active biomolecules and therapeutic proteins to create a robust anti-tumor immune response. We spoke to Howard Davis, CEO of Akamis Bio, about the challenges immunotherapies have faced in addressing solid tumors, the company's tumor gene therapy platform, and how it turns tumors against themselves to stimulate an immune response. Howard, thanks for joining us.
Thanks very much, Danny. It's a pleasure to be with you, and I look forward to the conversation.
We're going to talk about Akamis Bio, its platform technology that turns solid tumors into drug factories, and how this is designed to elicit an immune response to attack and clear tumors. Perhaps we can start with the need. We've seen great advances in immunotherapies, but the success to date has been focused on liquid tumors. Why has this been difficult to do in solid tumors?
Yeah, it's a great question, Danny. And at the heart of it really are some of the mechanisms that solid tumors have evolved to really be able to mask themselves from the immune system. And so, when you look at the tumor microenvironment, the fact that you've got expression locally within the solid tumor of immunosuppressive signaling molecules, you have significant amounts of stroma that these tumors encapsulate themselves within that prevents access to these tumors by the immune system. There's a hypoxic environment, high intratumoral pressure. All of these things contribute to an environment that makes it very difficult for the immune system actually to even recognize the tumor, much less attack it and try to clear it. So again, a number of just innate challenges for immunotherapy to be effective against solid tumors.
What's the need and opportunity to apply immunotherapies to solid tumors?
Yeah, so really what has been needed for some time to enable immunotherapy in this space are therapeutics or approaches that allow you to alter that tumor microenvironment. So some of those same defenses that tumors have evolved to be able to mask themselves, if you can reverse some of those effects or counteract them, you've got an ability to allow the immune system to recognize that tumor, attack it, and obviously the potential to clear it. And so that very much is what we are focused on here at Akamis Bio with our platform to really try to unlock that opportunity given the high unmet need for treatments of this type for the solid tumor space.
You joined the company in 2022 as CEO. What attracted you to Akamis? Why'd you take the job?
Yeah. I think for me, in the early stage biotech world, first and foremost, it's really critical that you believe in and are excited by the science. And that certainly is squarely what is here at Akamis Bio. The platform, which we'll talk a little bit more about shortly, came out of the University of Oxford. The team that's been in place here has put forward a tremendous effort to bring that platform forward into the clinic. And it's generated some great clinical data. And so I think first and foremost, that's what got me excited about the company. I think, the combination of that with a very supportive set of private investors who have been supporting the company for some time. And frankly, a set of investors who had a vision to take what was an Oxford-based company and evolve it into a true transatlantic entity, which is what we are now. So I'm based in the Cambridge, Massachusetts area here in the U.S. US, but we've also got significant operations still there in Oxford. And so getting the best of both worlds from both ecosystems.
Akamis has developed a platform it calls T-Sign. What is T-Sign?
Yeah, so T-Sign is an acronym for tumor-specific immunogene therapy. So we're a tumor gene therapy whose objective is to use a viral vector to alter the tumor microenvironment in solid tumors. And so specifically in our case, we use a chimeric adenovirus-based vector. It's been engineered to replicate and express a therapeutic transgene selectively in epithelial-derived tumor cells. And what's unique about this particular platform relative to the broader field, so by very nature, we're what is called an oncolytic virus, so a virus that has a tropism to replicate within a tumor cell. And in our case, our T-Sign platform has been engineered specifically to replicate and express those transgenes after systemic delivery. So we deliver our therapeutics in the clinic via an intravenous injection, which enables the virus to leverage the vasculature to basically transit to a tumor site where the virus can then replicate in either primary or metastatic tumor, where it then expresses a transgene that brings the immune system to bear. This is in contrast to the rest of the field that really has always depended on what is called intratumoral injection, where you actually have to physically inject into each tumor site. So this ability to deliver systemically is a unique aspect of T-Sign, and one that has us quite excited by some of the data that we've generated with it in the clinic to date.
How specific are these viral vectors to various tumor types, and do you need to do something to target them, or do you need to select the vector based on the tumor?
Yeah. So, one of the things that is so attractive about T-Sign is that, we are leveraging an engineered adenoviral vector to specifically target the tumor. But the characteristics of the vector itself are what drives the specificity for its both recognition, replication within, and expression of the therapeutic transgene specifically in the tumor. And so, our vectors basically have a tropism that allows them to replicate in any epithelial-derived tumor cell. So if you think about pancreatic cancer, colorectal cancer, head and neck cancer, all of these are epithelial-derived solid tumor types. And once again, the characteristics of the virus itself are what provides for that selectivity to again replicate and express the therapeutic gene in those target tumor cell types.
You have the potential to use payloads that deliver a combination of transgenes. What are the range of things that might be in a payload that you would get tumor cells to express?
Yeah. So, again, what's so exciting about this platform, certainly for us as a company and for me as I was looking to join last year, was the flexibility of what you can put into that transgene cassette, or the payload, if you will, that our T-Sign vectors carry. And we've, over the years of our work with the platform, have expressed a wide range of molecule types, including membrane-bound receptors and ligands, full-length antibodies, cytokines and chemokines, bispecifics, as well as tumor-associated antigens. All of these types of molecules, when expressed locally within a tumor, allow you to alter the tumor microenvironment, either enriching a target for another type of therapy, such as if we were going to express some additional tumor-associated antigens on the surface of the tumor. We can heat up the tumor microenvironment in what otherwise might be a cold tumor, again, by locally expressing cytokines or chemokines very locally, directly within the heart of the tumor itself. So again, very flexible platform in terms of our ability to access a number of different types of biology based on what we choose to express. So again, very flexible in terms of what T-Sign can do into the tumor microenvironment.
How do you determine what combination of constituents you'll include in a therapy, and are you at all limited by the size of the vector?
Yeah. So, in terms of, and maybe I'll answer your latter question first. Our in our work with T sign vectors, we found that typically we can get up to about 3.5 kilobases of whatever it is that we want to express from a genetic perspective, loaded into a T sign therapeutic, and that can be a single protein. It can be multiple proteins. We've had up to 5 different proteins that are expressed in that single 3.5 kilobase cassette. Typically, we have in most of our clinical vectors, between 2 and 4 genes that typically are part of a package. In terms of how we select what's going to go in, it really comes down to the biology that we're looking to either activate or interrogate. And so, an example that I'll give that we'll talk some more about is, around our lead clinical stage program, which is called NG350A. It specifically is a vector that looks to agonize the CD40 pathway. And so, given that that's the biology that we're going after, in that particular case, we've selected basically an antibody that we express that allows us to hit that target. So really, in terms of choosing what we want to do, it's really thinking about what is the tumor type that we're looking to target, what biology or again, biological pathway are we looking to attack, and then that motivates what we choose to put into a vector. But the flexibility of this platform in T-Sign is quite phenomenal in terms of what you can put in. And again, it gives us a very broad range of tumor types that we can potentially go after over time with this platform.
And is it expected that these therapies would be used as monotherapies, or would they be used in combination with other immunotherapies or other cancer therapies?
Yeah, so certainly in our early stage clinical studies, we're exploring these in the monotherapy setting, primarily to, as you can imagine, initially establish safety in the clinical setting. We certainly imagine some of the biology that we're going to go after would enable these to be used in a monotherapy setting, given the very nature of cancer and cancer therapy as we know it today, where combinations of agents typically are the way that standard of care therapies or therapeutic approaches are put together. We would anticipate that we will for sure be used in combination with other agents that are standard of care elements of either solid tumor specific therapy or more broader cancer therapy. So checkpoint inhibitors and such, we certainly would be expected to be using these agents with. The other application, though, that I want to highlight, and this is something that we're quite excited about from a collaboration perspective, and we have some news on the horizon around a collaboration in exactly the space that I'll speak about, and that's really using T-Sign as a neoadjuvant approach to basically enhance either the safety or efficacy of other immuno-oncology agents. And by this, I mean we can pre-treat a solid tumor with a T-Sign vector that alters the tumor microenvironment in a way that makes it easier for another IO approach to again deliver its efficacy. So the example that we've recently had some collaborations consummated in that we'll shortly announce is in the cell therapy space, where our role really is to alter the surface of the solid tumor cells to enrich for a target that a cell therapy could then recognize. So we really like this approach because we think it's one that offers certainly collaboration opportunities for the company, but also the ability to enhance again the efficacy and potentially safety as well for other IO modalities or approaches.
Well, you made reference to your lead experimental therapy. What indications are you pursuing with it?
Yeah, so to date, all of our clinical studies have been phase 1A safety studies. So really, we're looking to obviously understand the pharmacokinetics, pharmacodynamics of our viral vectors, and obviously get a good understanding of the emerging safety profile. in those settings, we've had patient populations that basically are all comers from an epithelial-derived tumor perspective. So any patients with metastatic disease where they've got, again, solid tumors of epithelial origin. So we typically see a mix of colorectal cancer patients in these studies, pancreatic cancer, some head and neck cancer. And so at this point, again, we're really seeing that broad population to establish safety. As we get ready to move into our next phase of studies, particularly for our lead program, which again, we'll talk about in a moment, NG350A, that's where we're starting to look at where have we seen data in those all-comers populations that's starting to motivate us to select an expansion cohort where we'll have a single indication. And we do have some interesting hints that are leading us to prioritize some indications that we'll announce probably early in 2024.
How does it work?
Yeah, so NG350A, just to speak on that lead clinical program, it's a vector. So again, uses the same adenoviral backbone as all of our T-sign vectors, but in this case, the transgene cassette holds two genes that combine once they're expressed to form a CD40 agonist monoclonal antibody. So again, given the characteristics of a T-sign therapeutic, replication of the vector only occurs within a solid tumor once following intravenous delivery. When that replication is occurring, you get local expression of that CD40 agonist monoclonal locally in the solid tumor microenvironment. The particular mechanism that we're looking to go after, so CD40 agonism is a well-known biology. It's been characterized via systemically delivered monoclonal antibodies of this sort. And in our case, we're looking to hit this pathway in the same fashion. What it does is trigger the dendritic cell pathway. So the dendritic cells that are typically present in the draining lymph nodes of solid tumors, which have the ability to present tumor-associated antigens to infiltrating T cells and then trigger basically an anti-tumor immune response, emanating from those draining lymph nodes. That's the pathway that we're looking to activate. And so CD40 agonists that are expressed in those draining lymph nodes trigger that dendritic cell antigen presentation, and again, the resulting immune attack on the solid tumor. So that's the pathway that we're looking at, and again, why we're so intrigued by NG350A. We believe that the ability to locally express that CD40 agonist monoclonal antibody directly in the tumor will allow us to hit that pathway much harder than you could if you were trying to systemically deliver the same CD40 agonist monoclonal antibody as a naked antibody alone. And that really is due to the fact that this pathway is well known to have dose-limiting toxicities associated with it when you deliver it systemically. And so because our T-sign vectors drive local expression in the tumor, again, we'll be able to hit that pathway that much harder by having much higher concentrations locally in the tumor. So that's really what we're going after, and we're quite excited by some of the early results that we've seen.
Well, what is known about it from the studies you've done to date?
So the NG350A program has been in basically a pair of clinical studies that we've run. The first was called Fortitude. This was a monotherapy study, classical dose escalation. Patient population, once again, as I described a little earlier, was all epithelial derived, all comers, I should say, from a patient population perspective, with epithelial derived solid tumors. We completed that study just under a year ago, and then have been moved forward into Fortify, which is also a dose escalation study, but in this case, it's NG350A in combination with pembrolizumab. So obviously looking at a PD-1. So those are really safety studies looking to get a good understanding of, obviously, again, PK, PD, as well as the safety profile of NG350A. In parallel with this, we also have a study ongoing that is in collaboration with the Parker Institute and Cancer Research Institute called Revolution. And this is an expansion cohort study in pancreatic cancer. And this will be our first chance to get a look at how does NG350A actually impact disease progression in a single patient population. So those are results that we'll be seeing in 2024, and certainly quite intrigued to see the impact on patients.
Well, talk a little bit about your partnership with the Parker Institute. How does it work, and what does Akamis get from this partnership?
Yeah. So the Parker Institute has been a long-term collaborator of ours. And, I would be remiss not to give a shout out of thanks to Sean Parker for his both commitment to the company and support of us, both in the form of this collaboration. And he's also, he and the Parker Institute are an investor directly in us. The relationship really started around NG350A and some excitement that the Parker Institute has had for some time around the concept of CD40 agonism. And so when they heard about our program and obviously a vector that would allow you to agonize CD40, but with local expression of an antibody in a tumor, they were quite intrigued to get involved and see how they could support us, in our development efforts. And so this particular collaboration, which is around the Revolution study that I mentioned, so it involves the Parker Institute is providing both funding as well as access to their clinical network, which are some of the sort of most esteemed pancreatic cancer investigators in the world. So they're supporting us on both of those fronts, financially as well as with access to that network that they've established. The Cancer Research Institute is also providing funding as well as operational support, and they've been a wonderful partner that we announced when we talked about that expanded partnership earlier this year. And then from our side, obviously, we're providing drug and then strategic insight into both the study design and execution. So, at the end of the day, we'll obviously get access to the data that emerges from that study. And our hope is that we see promising results next year that'll motivate us to advance that program into later stage studies in the pancreatic cancer space.
You're also approaching the clinic with a second experimental therapy. What is this, and have you identified indications you'll pursue with it?
Yeah, so the therapy you're referring to is something that we term NG7XX, and so this is it's a preclinical vector, so it's in IND enabling work right now. It's sort of two critical elements of the payload, if you will, are IL-12 and IL-15. There's also a chemokine that gets expressed in the transgene cassette. The objective of this vector is primarily to heat up the tumor microenvironment with the expression of again those interleukins along with that chemokine, and therefore to take what would ordinarily be a cold tumor and make it hot. In terms of the target indications, very similar to the way that we've developed NG350A, our plan would be, as we bring this through the remaining of its preclinical work over the course of the next year or so, we'll be readying the program for the clinic, where we will run likely a very similar type of monotherapy dose escalation study to start. We would then move, in all likelihood, into a combination study with PD-1s, and the patient population would again be all comers from an epithelial-derived tumor perspective. And again, what that affords us is the ability to accomplish our safety study objectives, which always are to understand PK, PD, and safety. But in the same token, we'll have an opportunity to assess a number of different solid tumor indication types that come into that all-comers population to determine where we want to prioritize as we would move into a later stage study. So no determination yet on exactly what indication to pursue, but likely a very similar type of development program to what we've taken with NG350A.
The company raised $30 million in January. How far will existing funding take you, and what's the plan for raising additional capital?
So we were really excited to announce that $30 million convertible note financing in January on two fronts. First and foremost, it was an opportunity to recognize three new investors that were joining us on the journey, and that was Arch Ventures, the Parker Institute for Cancer Immunotherapy, or PICE as they're known, as well as Westlake Village Bio Partners. In addition to being new investors in us, they are our first American-based or U.S.-based investors, which obviously coincides with my hiring in the Cambridge area and the transatlantic expansion of the company. And so that financing and our current financing will take us into 2024. We're currently actively financing right now to fund the expansion cohort study, which is our next stage of development from a clinical perspective for NG350A, that lead clinical program. So we'll be moving into a phase 1B study where we'll have a single tumor type that will be prioritized with an objective to demonstrate clinical proof of concept over the course of the next few years as we execute against that study.
You spent about 5 years each at Novartis and Biogen, and have seen how large biopharma companies operate, but you also spent a year at Flagship Pioneering. I'm wondering how that may have changed your view on the industry and on technology, and whether you're bringing anything from that experience to shape a company built on a novel platform technology.
No, it's a great question, and as I was being recruited into Akamis Bio, obviously it's a as you're being interviewed for a role like this one, it's a great opportunity to reflect on the career journey and what you've seen and how it shaped you. I think for me, as I look back to those experiences in bigger pharma and bigger biotech with Novartis and Biogen respectively, they were critical formative experiences for me in really learning sort of end to end how medicines are made. And again, lot is said about bigger pharma and bigger biotech, and some of it can tend to be pejorative around them being sort of slow lumbering machines. But at the end of the day, the bulk of the medicines that are out there today do come through the hallways of those places. And so it was a great place for me basically to apprentice and learn that process and learn how to think about unmet need and how you bring those things forward. I think as I pivoted to, what I'm going to call the more recent chapters of my career, and Flagship Pioneering was where I went first after leaving Biogen. And then, even post that, I spent a good period of time, nearly 4 or 5 years, working with the Atlas Venture folks on a number of different early stage company projects. And I think there's a skill set that comes from the flagships and the atlases of how do you build a company, and how do you build a company honestly from scratch in many of the environments that I've worked in? How do you build a management team? How do you secure the financing that you need to de-risk the platform technology that you're developing or the assets that you're bringing forward? And so, in many ways, as I look back at my career and the places I've been fortunate enough to work, the success that we're driving here, and the early success, I'll call it, certainly at Akamis Bio, and certainly the promise that this platform has, I owe a lot to both of those environments, both the big pharma and big biotech, as well as to what I've learned from working with in Atlas and Flagship, some very prolific creators of very valuable early stage companies. It's that combination that really I'm bringing to bear every day here at Akamis Bio.
Howard Davis, CEO of Akamis Bio. Howard, thanks so much for your time today.
Thanks very much, Danny. It's been a pleasure speaking with you.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.